All right. Hello, everyone. I'm Charlie Werth. I'm a program director here at ARPA-E. We have 4 great talks lined up for you today, giving our vision of things that we've been exploring here at the agency. We're going to cover destruction of atmospheric greenhouse gases, ammonia recovery from wastewater. We'll also be looking at plant engineering for climate and the future of carbon. And right now, I'm going to turn it over to our first speaker, Zach Berquist.
All right. Thank you, Charlie. Yes, so my name is Zachary Berquist, and I am a fellow at ARPA-E, and I am really excited for the opportunity today to talk to you about atmospheric greenhouse gas removal, its feasibility, and why we should do it. I'm coining this direct air destruction, or DAD, which means I have to start this talk with a dad joke. So, what do you call an unnaturally strong greenhouse gas? An HGHGHG. That is the appropriate amount of laughter for that joke. So, we need negative emissions technologies if we have any chance of meeting our climate goals. Most of this focus has been on carbon dioxide since it's 80% of global emissions. The rest of these, the majority, are methane and nitrous oxide. But I also want to highlight hydrogen. It's a very potent indirect greenhouse gas, so its effect actually isn't even accounted for in this plot. At some point this century, the net emissions of these other greenhouse gases will exceed that of CO2. They will become the bigger issue, yet they receive just 1% of global climate finance. So what I'm proposing is a concerted research and implementation effort for the negative emissions technologies for these gases so that we can help meet our net zero targets later this century. There's a key timing issue to these 3 gases specifically. First, for methane, as the planet warms, natural biological sinks, like wetlands and melting permafrost, their emissions are increasing. Their meat consumption is on the rise. As developing countries get wealthier, they eat more meat. This is a major source of nitrous oxide and methane emissions. And lastly, there's a booming hydrogen economy on the horizon. Hydrogen is quite literally the smallest gas molecule possible. We already deal with methane leaks, so we're inevitably going to have hydrogen leaks, and this will have a significant warming impact. I want to make note that ARPA-E has actually already been a thought leader in this space with our recently announced H2 Sense exploratory topic, though this is looking at hydrogen leaks for mitigation. And again, I'm looking at atmospheric removal. Removing any greenhouse gas is incredibly difficult because just 3 gases make up over 99.9% of our atmosphere. All of the rest of the gases, there's such a small fraction. Microsoft PowerPoint doesn't even render the slice on this pie chart. So if we zoom into this unrendered slice, the majority is carbon dioxide. And the gases that I'm concerned with are that smallest slice. So a technology that we might use for carbon dioxide, direct air capture, is actually not feasible. They're too dilute. But is there something else we can leverage? I'm calling these 3 gases the reactive greenhouse gases because they, compared to other species in the atmosphere, they have relatively short atmospheric lifetimes, and they eventually degrade the products that are less potent and much more stable. What's interesting, though, if you look across the atmosphere and you look at other species that have high chemical potentials, you'll find pollutants, things like black carbon and volatile organic compounds. They eventually degrade under the same mechanism, or similar mechanisms, and they also have high global warming potentials and again degrade to ones that do not. So to understand how we might do this, we can look up into the upper atmosphere and see how Mother Nature does it herself. High energy radiation splits water and oxygen molecules into their respective radicals. These are known as the atmospheric detergents. Closer to Earth's surface, this high energy radiation is attenuated and only slightly lower energy ultraviolet radiation makes it to the surface. It is only capable of splitting water. So hydroxide radicals near the Earth's surface are known as the tropospheric detergents. One side effect of the formation of these radicals is the formation of ozone. Up in the upper atmosphere, in the stratosphere, this is a good thing. Near Earth's surface, it is harmful. It is a harmful pollutant to both humans and plants, and it's a potent greenhouse gas. So if there's a way that we can make these atmospheric detergents without forming these other negative side effects, we can actually accelerate the degradation of these reactive greenhouse gases. So there's 4 solutions, or DADs, that I'm going to discuss. The first is building DAD, then HVAC DAD, ocean DAD, and microbe DAD. So for building DAD, people have previously proposed the idea of applying photocatalytic paints to buildings and roads, and these will passively generate these atmospheric detergents and start the reactions of eliminating these reactive greenhouse gases and other pollutants. But if you just apply these photocatalytic paints liberally and you just don't really control what's going on, you are inevitably going to make ozone, and that's something that we don't want to do. So can we come up with a better idea for this? What about photocatalytic concrete, something that can harness the power of sunlight, but we can transfer the high energy electrons that are formed into the depths of this porous, intelligently designed substrate, where we can actually control the reactions and selectively convert these reactive greenhouse gases. For HVAC DAD, one of the biggest costs, both operating and capital for direct air capture plants, is simply just their HVAC system, moving massive amounts of air so we can maximize contact with CO2 in the atmosphere. So we can just put these reactive gas destroyers on the outlet stream, leverage the sunk cost of these HVAC systems. We can control the environment in these single unit ops and hopefully mitigate the formation of negative byproducts, and again, maximize the conversion of these reactive greenhouse gases. But then I thought, why am I limiting it to DAC? We move massive amounts of air all across our economy in industrial and commercial settings. So we should be applying these reactive gas destroyers on the outlet streams of all of our systems that move massive amounts of air. So for ocean DAD, seawater is salty, and it occasionally gets kicked up into the lower atmosphere, known as sea spray. In a series of reactions with chlorine ions, iron-based particles, and acid, now this can be sulfuric or nitric acid, typically from flue gas, from ships or power plants, they will react together, eventually forming chlorine radicals. This is an atmospheric detergent. So even if it forms ozone, if you're doing this in the middle of the ocean, it's so far away from people and plants that its negative side effects are mitigated. And this chlorine radical can again kickstart many of the processes for the degradation of reactive greenhouse gases and other pollutants. And lastly, microbe DAD. Microbes are already the largest natural existing sink for hydrogen. It consumes 60% of the hydrogen today. For methane, it's much lower, but it still exists. It's like under 10%. But if we can increase the amount of these microbes, increase their reactivity, proliferate them, we could have a passive destruction of these gases. You will note that nitrous oxide is not on here. That's because nitrous oxide is formed in the soil. It's a completely different beast under there. I will say, if this is something that interests you, my colleagues Ben and Steve have already talked about this. They've already been thinking about this, so I would go talk to them. One thing I will leave you with is that there is at least one biological pathway that actually converts nitrous oxide back to nitrates and ammonia. So, I know you're all clamoring for more dad jokes, so I put 4 more up there. I'll just wait for enough laughter before I move on. Yep. So if any of these dads excite you, please come talk to me at the coffee hour tomorrow or email me, zachary.berquist@hq.doe.gov. Thank you. Now I'll hand it back to Charlie.
All right, thanks, Zach. So in the spirit of dad jokes and in a transition to my own topic, what do you call someone who's a wastewater sewer expert? Zach? No?
I don't know.
A connoisseur. All right. So it's great to be here today. I'm going to talk about ammonia recovery from wastewater as an energy resource, perhaps not the most comfortable topic. We often think that wastewater is kind of gross. and we don't like to think about our waste when we flush the toilet. What happens to it? Well, my aim today is to convince you otherwise. What I mean is, my aim is to convince you that wastewater is beautiful, all right? Not aesthetically, I get it, but it's beautiful because it contains ammonia, a lot of it, all right? And if we can recover that ammonia, we can reduce by 40% the amount of ammonia we need to recover from the Haber-Bosch process. However, for this to happen, we really need a transformational change in materials and processes for this recovery. This change, I think I went 2 slides back. There we go. All right. This change is needed because we expect the demand for ammonia to grow 50% over the next 30 years. A lot of this will be used for agriculture. That's where most of it goes now. But we'll also need it for transitioning to the hydrogen economy. When we think about the hydrogen economy, we expect to use renewables, that off-peak energy, where we have excess, convert it to hydrogen. We anticipate converting that to ammonia as a more dense energy carrier. We also anticipate using ammonia as possibly a maritime shipping fuel. We really have an incredible opportunity to recover and save energy and to reduce greenhouse gas emissions, but new ideas are needed. Each year, we use 1% of the Earth's energy to make ammonia through the Haber-Bosch process. This is a plant shown here on the left. Through steam reforming of methane, we create hydrogen, and then combine that with dinitrogen at high temperature over a catalyst to create ammonia. That process emits more than 450 million metric tons of CO2 each year. We justify that energy demand and those greenhouse gas emissions because ammonia, as fertilizer, is the lifeblood of crops that feed the world. If you look on the right, as we apply more fertilizer to farm fields as nitrogen or a nitrogen source, which comes from ammonia, we get an increase in that corn yield, up to a limiting point, of course. Perhaps fortunately, almost half the ammonia that we apply to farm fields ends up in wastewater. That's human, animal, and industrial wastewater. In the US, that comes to about 6 million metric tons of ammonia of the more than 13 million metric tons of nitrogen that we apply to crops each year. This creates an untapped potential to recover 40% of that ammonia that we make with the Haber-Bosch process. In the United States, that would yield an energy savings of about 0.4 quads per year, or worldwide about 10 times that amount. With 4 quads of energy, we could power the entire state of Illinois and then some. This also creates an untapped opportunity to reduce greenhouse gas emissions associated with wastewater treatment, mainly in the form of nitrous oxide emissions, which I'll elaborate on the next slide. If we could reduce those greenhouse gas emissions as nitrous oxide, and then eliminate the CO2 from that 40% of the Haber-Bosch process producing ammonia, we could reduce greenhouse gas emissions by more than 1%. I'm going to try it again. There we go. All right, this slide's a little busy, but it represents the state of practice in wastewater engineering. And so if you look at this, we have an influent and effluent, and we have a simple screening and then gravity settling. But the main star here, the main process, is activated sludge. We put energy into that for aeration to destroy the organic waste and also to destroy the ammonia. When you look at the total organic carbon, or TOC, it shows that we do a really good job of destroying the organic waste. It goes from 200 in the influent to 20 milligrams per liter in the effluent. We also do a great job of destroying the ammonia. It goes from 25 to 0.4 milligrams per liter. But we're only converting that ammonia to nitrate. And when you look at the nitrate, we don't have any coming in, and it goes up to 26 milligrams per liter. Nitrate, or nitrogen, is often a limiting nutrient in surface waters. And so what ends up happening is we have eutrophication, hypoxia, damaged ecosystems, and really just icky-looking surface waters when you look around. Just when you thought the problem couldn't get any worse, we have a whack-a-mole problem, right? We try to take care of ammonia by converting it to nitrate. Sometimes we convert that nitrate to dinitrogen in a plant, but oftentimes it happens in the environment. And what happens are those large nitrous oxide emissions. Nitrous oxide's a really nasty greenhouse gas. It has a global warming potential that's almost 300 times that of carbon dioxide. As a result, we're putting more than 40 million metric tons of CO2 equivalent into the air, and that has an impact that's about 3 times greater than the CO2 emissions from the equivalent ammonia that we're not producing. Really, all these impacts make it clear that we need to change the paradigm of wastewater treatment. We can't think of these feces and urine and other industrial chemicals as nasty products. They're really a commodity that we have to recover and put back into the supply chain. The barrier to that is new materials and processes that selectively recover that ammonium ion or the ammonium gas in a complex wastewater matrix. Luckily, we've made great advances in recovering other ions and gases, not ammonia, right, from other streams. So for example, if you look at the left, here we're showing that there's been really great advancements developing membranes with narrow pore size distributions where you can separate out one gas from others in a mixture. If you look at the middle figure, we've been developing really selective functional groups that can chelate specific rare earth elements over others, maybe in an adsorbent or in a membrane. And then if you look at the right, in terms of electrochemical processes, there's been advancements looking at membraneless electrochemical processes or using non-precious metals, for example, for water splitting to create hydrogen. The challenge is that these insights, or the insights gained from these advancements, haven't been adequately translated over to looking at ammonia recovery, especially when we have this complex wastewater matrix. We really need to take advantages of these properties of the ammonium ion and ammonia to selectively recover it. So for example, on the left plot, it shows the ammonium ion and how it differs in size from other ions that you would expect to find it with in solution. And that could be either in the hydrated or the non-hydrated form. The middle plot shows how we have ammonia, which is a hard base, ammonium ions, a soft acid, and how we can use those concepts as a surrogate for these electron energy levels and molecular orbitals to identify really selective functional groups that we can put into adsorbents and membranes and other materials to separate them. And then the right plot shows how, with knowing the proper reduction potential to apply, we can selectively oxidize ammonia and create hydrogen. Ammonia is a really valuable resource. It has a high cost. It also fluctuates because it so depends on the price of natural gas. We expect its use to go up by 50% over the next 30 years. We expect to use it either directly or after conversion to hydrogen to power engines for electricity or maritime shipping. It's also likely to remain the #1 agricultural amendment in order to feed a growing population with decreasing agricultural acreage. We really need new advances to address this issue and to recover this valuable resources from this complex matrix. If you're interested in ammonia recovery from wastewater or critical minerals recovery, please come talk to me, or you can contact me at this email address. Also, we just had an RFI come out looking at recovery of high-value materials from wastewater. Thank you. Ben, take it over.
Thank you very much, Charlie, and thank you all for being here. I apologize in advance for the lack of dad jokes, but we will soldier on. So my name is Ben Weiner, and today I'm excited to talk about plants as a climate tech platform. So this is important because the world has a carbon problem. Each year, human activity releases about 40 gigatons of CO2 into the atmosphere. Unfortunately, climate models suggest that if we want to limit warming to 2 degrees Celsius, we need to not only achieve net zero, but then remove about 50 gigatons from the atmosphere by 2050 and sequester it either in stable materials or geological reservoirs. And to reach net zero, we'll probably need to incorporate captured CO2 into less stable products as well. And so the question is, how are we going to get all of that carbon out of the atmosphere? Well, planetary problems require planetary solutions. And so we'll need to harness the power of chemical engineering through direct air capture, the power of geology through enhanced weathering, and perhaps even the power of the oceans through various forms of ocean capture. But a complementary approach that I'm really excited about, which you probably could have guessed from the title of my talk, is to use plants. So plants are a really powerful platform for climate for a few reasons. And the first is they get their energy directly from the sun, unlike direct air capture, which requires heat or electricity. The second is that they're extremely scalable. Because we have a global agricultural system, we can distribute seeds to tens of millions of hectares in a single season with very little additional capex. And the third is that unlike enhanced weathering or ocean capture, plants produce valuable goods, which can finance carbon removal while providing for our societal needs. But the disadvantage is that plants can place great stress on our land, water, and fertilizer systems. And so plants have this unique scalability, which makes them the software of biotech. But if we want them to play a sustainable role in carbon removal, the software needs an update. So let's imagine a software update for plants. First, you'd want to enhance photosynthesis and introduce nitrogen fixation to alleviate land use constraints and fertilizer demand. Then you'd want to optimize the root system to restore soil health while seeking out water and nutrients. Perhaps your plant has seeds which produce oils that are tuned for sustainable aviation fuel, while the stock accumulates polymer precursors that can be turned into sustainable plastics. And of course, you want it to be resilient to extreme weather and pathogens. And so there are scientists all over the country, some of whom referenced here, who are making great progress on all of these traits. And if we could implement them at scale, it would be transformational for agriculture, of course, but also for carbon capture and utilization, for bioenergy, for plastics and materials, and for our very stewardship of the land and soil. But if we want to take these discoveries out of the lab and into the field, we need to get much better at plant engineering. So conceptually, plant engineering is similar to software engineering. For software, we write code using a text editor and then compile it into an application. For plants, we edit the genetic code in a single cell using bacteria as a text editor in a process called transformation. And then in a process called regeneration, we grow that plant cell into a whole organism. So just as for software, we have coding, compiling, application. For plants, we have transformation, regeneration, organism. But there's a catch, because plant engineering is way harder than software engineering. And there are a couple reasons for that. The first is that you're inheriting the world's most convoluted code base. Plant genomes have been shaped by hundreds of millions of years of stochastic evolution, and we still don't know what most of that DNA does. The second challenge is that there's no general text editor. These text editors are microbes, and in the same way that microbes can affect each of us differently, they don't generalize across plant breeds, much less species. And in fact, we still don't have text editors for many of the crops that we care most about. The third challenge is that we can still really only edit a line or two of DNA at a time, which prevents us from making more systemic changes. And finally, this compiling step of regeneration takes more than 9 months to complete, which prevents us from rapidly iterating. And to be honest, regeneration remains a pretty fussy and artisanal process. So the folks with the magic touch for this are in very high demand in academia and industry. And so as a result of these challenges, even at the big ag companies with all of their resources, it takes 8 years and $70 million to bring a new trait to the regulatory stage. And if we've learned anything from the world of software, it's the power of a rapid design, build, test loop. And so it's clear that this is going to severely limit innovation. So these challenges are no secret in the field, and there's been real progress recently. For example, new CRISPR-based tools, which allow us to edit the genome with increased precision, and a better understanding of the morphogenic genes controlling development holds the promise of more reliable regeneration. But frankly, this is incremental progress, and if we want to transform plant biology on a timescale that matters for climate, we need to accelerate. So I'm going to make 2 proposals to accelerate the design, build, test loop for plants from years to months. And the first is to fix the text editors and skip regeneration altogether. So in the language of software, this would be like moving from a compiled language, such as C, to a scripting language, such as Python. And we know that this is possible because Arabidopsis already does it, making it the Python of plants. Now, this humble weed has become the workhorse of plant biology because you can just dip the flowers into a suspension of agrobacterium, and it will produce a seed that grows into an organism. No regeneration and no tissue culture required. And the secret of this special relationship between Arabidopsis and agrobacterium, it's not really understood. We discovered it about 40 years ago, and we've been using it ever since without diving into how it works or how to improve it. And so I propose that instead of just taking the tools that nature's given us, we engineer our own. And in particular, we can understand the plant as a host using the tools of comparative genomics and proteomics, and then use synthetic biology to tweak the microbes in order to engineer the host-microbe interaction to make it general across plants and regeneration-free. Now, unfortunately, even if we succeeded at this, we'd still only be editing a line or 2 of DNA at a time. And so my second proposal is instead of just editing DNA, we should start writing our own. Now, this might sound like science fiction, but in fact, it's been possible to chemically synthesize DNA in the lab ever since the 1970s. And what I'm plotting here is the length of synthetic DNA that people have been able to make over time. And what you can see is that that very first piece of synthetic DNA back in 1977 was only 42 base pairs long. You can't really do much with 42 base pairs. But in the intervening decades, there have been orders of magnitude of progress, culminating in a 2019 paper where researchers synthesized an entire E. coli genome of 4 million base pairs. And so if you look at the scale here, we haven't yet reached the size of the smallest natural maize chromosome, much less a full genome. But we can make pieces of DNA containing hundreds of eukaryotic genes. And so as DNA synthesis continues to increase exponentially, it raises the question, can we synthesize artificial plant chromosomes with hundreds of genes at a time? Because if we could, it would revolutionize plant engineering. And so speaking of revolutions, if you take a step back, plants have already changed the course of human history twice. The first time in 10,000 BC, when the invention of agriculture led to the rise of civilization, and again in the 1960s, when the Green Revolution unleashed unprecedented productivity and prosperity. I believe that soon, plants could enable a third agricultural revolution, the transition to a sustainable and carbon-negative society, but only if we get much better at engineering them. And so if you're excited about this vision of plants as a platform for climate, or you have ideas about how to accelerate the plant design build test loop from years to months, then please come and find me at the coffee hour tomorrow or reach out over email. Thank you very much for your attention. Now I'll hand it over to my colleague, Steve.
So I'm Steve Singer, and I'm going to tell you today about the carbon transition. So decarbonization is a word that describes the energy transition from fossil fuel to low carbon energy. If we're really successful at decarbonization, we will use 80% less fossil fuel in 2050 than we do today. However, we have to remember that fossil carbon is not only used for fuel, but for the polymers and materials that are fundamental to our life and economy. Really, fossil carbon is all around us. It's in our kitchens, it's in our houses, and it's in our clothes. And we need to continue making and improving these products. How do we do that in an era of declining fossil fuel use? So to illustrate this, I want to show what we do with a barrel of oil. So now 85% of a barrel of oil goes to fuel, and 15% goes to non-fuel uses, including making precursors for polymers and materials. Now, as the use of that 85% declines, what are we going to do with this 15%? Where are we going to get it from? So another way to think about this is what I call the electric car paradox. Now, this electric car, we like cars at ARPA-E, if you listen to Hallie's talk in the last session. So this is a BMW i3. It was designed about 10 years ago. And this was a pioneering electric car. And one of the reasons it was a pioneering electric car, oh, of course. One of the reasons it was a pioneering electric car was its body was made entirely of carbon fiber. Now think about this. The electric car doesn't require fossil fuel. The carbon fiber body is entirely made out of fossil fuels. So if you think about that, therefore we need to go through a carbon transition that's analogous to our energy transition. And I'm going to focus on two approaches to this carbon transition, thinking about refining. And the first will be retooling fossil refineries to lower the amount of greenhouse gases produced. And the second will be to transform plants into living refineries. So I'll illustrate how we can go through this carbon transition using the carbon fiber in the body of this BMW and how we can make it in the future. So thinking about retooling the refinery, the reason we want to do this is that the operations of fossil refineries consume 4% of the total greenhouse gas emissions in the United States. And one of the most significant sources of these emissions is fractionation of crude oil. So on the left, you see, normally when you fractionate crude oil, use a cracker, a thermal cracker. However, what we can do now is we can go from thermal cracking to electrical cracking, which will lower the CO2 emissions. more broadly, we can power fossil refineries with renewable energy. So more innovations like this are really needed to lower the carbon intensity of fossil refineries. So in these retooled fossil refineries, we can make new carbon materials. So if you take hydrocarbons, like methane, and pyrolyze them in the absence of oxygen, you get hydrogen, but you also get solid carbon. And now that solid carbon can be spun into carbon fibers that can make the body of the BMW with lower energy and lower emissions than we do today. You know, but wait, I think many of you will say, we don't want to do this carbon transition with fossil carbon. We want to move to a carbon negative process. And for that, we can use plants. And now Ben has really eloquently described the extraordinary opportunities we have in engineering plants. So plants can take sunlight and CO2 and make complex carbon structures that are precursors to polymers and materials, making them living refineries. So to think about plants as living refineries, what we need to do is think about the current biorefinery model for plants. So in the current biorefinery model, plants are used as sugar sources, which are extracted from the plant, and then through microbial, sometimes chemical catalysis, you can make an intermediate like muconic acid. And now muconic acid, through catalysis and polymerization, can be transformed to nylon fiber. So if we take plants as living refineries, in this new model, instead of going through this multi-step process, we can make an intermediate like muconic acid directly in the plant and then use the same catalysis and polymerization steps that we use for the microbially produced product to make nylon fiber. So why would we want to do that? Well, here's why. So taking muconic acid as the example. So you can reasonably get about a 50% yield of muconic acid from a sugar-based process where you use a microbe. So to achieve cost parity, you only need to produce 0.3% by weight in a plant. So what we can do here is really take advantage of the scale and efficiency of agriculture to really enable plants as living refineries. So let's go back to my favorite example of the BMW. So now it turns out that a precursor for carbon fiber is acrylonitrile. And acrylonitrile can be made from a bio-based intermediate, hydroxypropionic acid. Now today, hydroxypropionic acid is made from plant sugars through a microbially mediated process. But in the future, if we make plants living refineries, we can actually make plant-based carbon fiber. And that truly will be a carbon transition. Now, the reason we're interested in carbon fiber is not just for a 10-year-old BMW. Why we're interested in carbon fiber is if we can move from these low volume, high value products like the BMW to large volume, low cost products like building materials, we can then store gigatons of carbon in the built environment. So finally, I invite you to come talk to me after the session or at Coffee with ARPA-E about the carbon transition, our vision of carbon in a decarbonized world.
All right. Thanks, y'all, very much. I think we'll start off with Zach here. We have a question from the audience. Regarding the percentages of greenhouse gases, why don't we instead increase the fractions of nitrogen and oxygen, which would lower the relative percentages of greenhouse gases?
That seems to be geoengineering on a scale beyond anything we've ever considered. Yep. I don't know how the world, well, well, we can't really increase oxygen and nitrogen because they're already at 99 or 98%. So I guess we can increase oxygen, but then I don't know. I'm not a geoengineer. I'm not a plant or I'm not an atmospheric scientist. So
All right.
That's
Thanks, Zach. All right. Ben, how does the time frame to genetically engineer plants compare to using selective breeding?
Yeah, that's a great question. With selective breeding, you're limited to the generation time of your crops, although the big ag companies are pretty good at doing so-called speed breeding to accelerate this. But I would say the fundamental limitation of selective breeding is that you are stuck with the genetic diversity that already exists in the population of plants you're working with. Whereas with these engineering techniques I'm talking about, you can introduce entirely new things. So instead of just optimizing incrementally, you can have entirely new pathways.
Steve, did you have anything to add to that? I know you've been thinking about that.
Yeah, well, I think if you can, I mean, what I think breeding and targeted gene editing can kind of go together. You can breed and get the traits you want, but of course they're really complicated. But if you then look at the genomes, you can actually pinpoint where the edits are, and then you can go back in and make a specific edit. And so you can kind of get rid of a lot of the other the other alterations that you have in the genome.
Okay. Steve, how do you see CO2 conversion into chemicals, polymers, as fitting into this carbon transition?
So I think there has been a lot of work funded by the Department of Energy and other organizations about how to take CO2 and chemically or microbially convert it to chemicals and materials, including programs, current and future programs at ARPA-E like EcoSynBio and Greenwells. But I think plants as direct converters of CO2 are underappreciated. So really, that's what I want to think about. How can the direct conversion of CO2 to chemicals in a plant change how we think about the overall conversion?
Question for me here. Let's see. How do you limit biological variability within the wastewater treatment plant or in the sewage system? I think usually when we think about ammonia recovery, when we enter into that activated sludge system, the retention time is typically long enough. So first we're using up the carbon, and most of the nitrogen that the bacteria need is coming from those solids. And then we start using that ammonia. And the evidence for that is when we look at the effluent, most of that ammonia goes to nitrate, which is not incorporated into cells. And so I don't think we have to try to limit the microbial diversity or variability within a wastewater treatment plant. It's often, I think, self-selecting for that process. But we have to manage how we operate it and the retention times and the solids retention time as well, so that we get the carbon destroyed or the waste, but we retain that ammonia so we can pull that out. All right, let's see. Zach, what do you think are some of the unintended consequences of the technologies you proposed?
Yeah, it's a good question. So one of them, didn't really have time to touch on it in the 8-minute talk, but if you partially oxidize volatile organic compounds, you can actually make things that are more harmful than the original volatile organic compound. So if you're going to start the chain reaction, you have to complete it, which is why we really need better control with these reactions. And then the other is, I think the one that I personally find most appealing is the iron salt aerosol. That was the ocean dad.
Yeah.
You can actually increase methane initially. So there's like a lot of uncertainty. So you might actually have to hit a certain amount of these iron salt aerosols that you put like into the fuels of ships or power plants or anything to actually get over that hump and start the methane mitigation. So there's a lot of uncertainty, but that's why we just need to do more research on them.
Okay.
Yeah.
Ben, it's kind of a similar question we got for you. When you think about genetically engineering plants and you think of unintended consequences, there's often a lot of societal, concerns that come up with that. I wonder if you could address that.
Yeah, that's a great question. I mean, you want to prevent edited plants from escaping into the environment and proliferating. I think that people underestimate how hard it is to keep plants alive and really help them thrive. And I think, in fact, your challenge is more, optimizing, making sure that you're getting what you want out of them versus uncontrolled release. But there are important biocontrol steps that you can take. For example, a lot of GMO crops do not produce seeds for this reason, so that they can't spread into other fields.
Thanks, Ben. Steve, I don't know if that question applies to your program as well. When you think about new pathways, and are there risks there that are unique from what maybe Zach or Ben talked about?
Yeah, I think one of the obvious opportunities is there's about 380 million metric tons of corn biomass in the United States. So to actually make the volume of chemicals that we would need in corn biomass, we wouldn't actually need very much of the corn biomass to do it. I mean, maybe 1/10 to make all the muconic acid we would need for making nylon fiber. But if you think about that, it's a food crop, so we would have to make sure it's regulated so that the muconic acid stays in the biomass and does not move to the grain that we would use for, it's mostly for animal feed. But so, we can think about moving to non-food crops, but then we have land use issues. Where are we going to grow these non-food crops? Are they going to, replace food crops, or are we going to find new places to grow crops? I should say the idea of making products in plants is really compatible with both converting plant biomass to biofuels, the rest of the plant, or taking plants and using them as CO2 sequestration devices. Essentially, taking the biomass of the plant and trying to sequester it in the ground, either directly or through some chemical method.
Okay. Yeah. Thanks, Steve. Here's a question for me. Gas and liquid chromatography are often very selective and commonly used in small scales in laboratories for selective isolation of chemicals in solution. Is it possible to scale up these procedures to apply to wastewater in effective ways? And that's a really good question. We often think of chromatography in separating very small volumes of liquid or species within the liquid. When you have chromatography, you think about having an adsorbent, right? Some selective adsorbent or adsorbent which at least has different selectivities for the different compounds which are in that liquid. And so as your liquid moves through, right, the different components in that liquid separate out and they come out at different times at the end of that column. When you think about the ammonium ion, we have ion exchange resins which adsorbed ammonia in addition to other ions and with different affinities. And so certainly, I think from a theoretically or technical perspective, you could use chromatography to separate out ammonium from other ions in solution. Wastewater presents its own set of challenges. There's so much gunk in wastewater. When you think of it, you just don't have those ions in a pristine water. You have all sorts of natural organic matter with all different molecular weights and all different charges. And if you think about chromatography with natural organic matter, it comes out everywhere and just kind of increases your baseline. And so I think you'd have to do some pre-kind of cleaning step to remove that background organic matter so that you could then separate out the ammonium in a relatively clean form from those other ions. In doing that, you also have a scale-up issue then or challenge. We're used to putting through small volumes, and so you'd have to then design and operate a system that could put through, in parallel, probably, large volumes of liquid in batches, right? One batch after another, so you could collect that fraction coming out. So I guess the short answer is yes, it's certainly possible. I don't know how the economics would work out. Perhaps that's something that someone will want to look into and then cost that out and see if it's competitive. But it's really a nice idea. So thanks. Let's see. Ben, question for you. What specific plants would be the highest priority for synthetic chromosome engineering?
Corn is always an appealing target because we make a lot of it and we understand its genome relatively well, although there's still a lot of sort of genetic dark matter there. The DOE is also very excited about specific energy or carbon crops, such as switchgrass, miscanthus, crops which grow very rapidly, can hopefully be grown on marginal lands that wouldn't be used for food production as well. So I think that the nice thing about a lot of the tools that I'm talking about is that they would be pretty generalizable. You know, we want to build platforms that people can apply to the crop or the product or the application that is going to make the highest impact for them.
Thanks, Ben. Steve, this is kind of a follow-up to, I think you addressed it partially in your last response, but it has to do with, is there enough usable land to enable the carbon transition while not interfering with food agriculture?
I think there is, because I don't think we think that plants are the only answer. Because I think the idea of the carbon transition is that you need to take carbon from many different places, from fossil sources, from plants, from CO2. And I think what we want to do is try to think about an integrated strategy for making these carbon products. Now, if you think about the volume of the 15% I talked about, there's about 200 million metric tons of carbon that I think we need. And so, you know, I think agriculture actually can make up a good fraction of that with current usable land if we find clever ways to do it in current crops.
Zach, how can we know how much methane these technologies can pull out of the air? And also in terms of kind of monitoring that, is there an accurate way that we can, you know, keep tabs on it?
Yeah, I mean, measurement, reporting, verification, or MRV is going to be probably the single hardest issue, because then we also don't know how to value these technologies if we don't know how much methane we're actually pulling out. I think one of the, you know, we would have to build confidence in some of these modeling, but we could also have sensors that kind of sample from a large surface area to try to understand what's going on in the whole. So for example, I mean, if you're using the direct air capture method, that's going to be pretty simple to measure what's going on. If you're doing something like the photocatalytic paints or anything that's going on in the ocean or in the soil, you would just need a large array of sensors that could give us confidence with the amount of methane and nitrous oxide and hydrogen. So the H2 Sense exploratory topic, trying to come up with hydrogen sensors would be key for this. So yeah, definitely need innovations in sensing technologies. We actually have already done that with some of the programs already at ARPA-E. But again, kind of building these models and understanding flow patterns and everything would help as well.
Thanks. Question for me. What are the most promising scalable methods of recovering ammonia from wastewater? That's a great question. There's been quite a few technologies that have been evaluated in the lab, and a few that are even seeing their way into small-scale commercial applications. There's two companies I'm aware of that are looking at first absorbing the ammonium ion out of a wastewater matrix so that can be put into a cleaner solution, and then using electrochemical methods to oxidize the ammonia and couple that with hydrogen production. There are also, if you go back and look at kind of recovery of ammonia from kind of high concentration waste streams, there's ammonia stripping towers that have been used for a long time. At least conventionally, they haven't been as promising in terms of the economics for looking at low strength ammonia streams when we think about the wastewaters that I refer to. When you're thinking of ammonia stripping tower, usually then you have to adjust the pH, right? You have to increase it, so you go from the ammonium ion to NH3, and it can be volatilized. Also, if the water has quite a bit of kind of buffering capacity, which it usually does, then you have to also get rid of that as well. And so there's some expenses associated with it. There have been quite a few studies looking at selective adsorbents and membranes. In some cases, the membranes were combined with electrodialysis type systems for separation. I think those systems certainly hold promise. I think there are certainly challenges, the biggest challenges perhaps associated with fouling and how you can easily reverse that. You know, are there electrochemical methods to reverse it? Are there kind of easy chemical approaches to reverse that. And so I think they're certainly very promising, but there's going to have to be some good ideas to advance those. Ben, I have a question here for you. Is there a good DNA structure to property mapping to allow for genetic engineering? I don't know if I quite understand that. So do you understand the question?
Yeah,
Okay.
There's been a lot of, one of the big questions in biology is how does a particular sequence of DNA map to a particular protein molecule that does something, right? And the protein structure problem is a long-standing one. There's been a lot of progress recently with machine learning. You might have heard of AlphaFold, a great project out of DeepMind. We can do a pretty good job predicting structure now, and we can do a decent job understanding what those structures do. But I would say that it's very limited in understanding these more complex traits that emerge over the course of the life, the life cycle of a plant and its development, right? So you might know what this DNA makes this molecule, this makes this, and they bind together. But putting that into the full life cycle is really hard. So I think that these tools for understanding protein structure can play a great role, especially in discovery, in forming hypotheses to test. But I think that the structure problem, it only gives you things to try. You still need to then go in the lab and do it.
Zach, question for you. Does your analysis consider the effect of CO2 produced from direct conversion?
Yeah, so we're still making greenhouse gas when we convert methane to CO2. The thing, though, is that methane is so much more potent. It's got a GWP of about 80 on a 20-year time scale and 30 on a 100-year. But near-term warming is more significant than long-term warming because there's these positive feedback loops. It's more critical to stop warming today than it is 100 years from now because we'll just be more technologically advanced, more capable to address it then. So yeah, there's a huge motivation to convert methane to CO2, even if CO2 itself is still a greenhouse gas, because again, it is just so much less potent.
Yeah. Thanks. I have a question here for both Steve and Ben, and we'll let Steve address it first. Do you think using plants for making commodity chemicals will require more energy for extraction and downstream processing? And would direct carbon capture via microbes be better?
I think direct carbon capture for microbes is going to be very difficult. So if you think about the surface area that you need to capture CO2, it's going to be the plant. Plants have optimized for capturing CO2, in the environment, and humans have actually optimized it. I think microbial CO2 is going to be very difficult. But so what I will say also that if you compare microbial production to plant extraction, if you think about building biorefineries, if you're building biorefineries just to extract a product rather than to convert microbially sugar to a product, I think they'll be easier to build, they'll be cheaper. So they will look more like ethanol plants and could be done at an ethanol plant. I think one of the problems with scaling the microbial conversion has been that you need sterile environments for a lot of these production. You need large facilities, and it's been very difficult, especially at scale, to build those facilities.
Thanks. Ben, maybe I'll modify the question a little bit for you, but I think maybe the same thing would apply. So when we think about making or modifying plants to increase carbon uptake, is there any sort of downstream kind of energy requirement in terms of processing that would be greater than, I don't know, maybe you didn't consider or account for?
For many of these processes, you're going to need to do downstream processing. There are genetic traits you can go after that ease that. For example, there's been work on engineering plants or breeding them to have fewer lignin molecules, which are hard to break down, and so that makes the sugars more accessible. So you can imagine doing things like that. But if you get really good at gene editing, sort of your creativity is the limit, and you can think about other ways of extraction. So for example, many years ago, there was an ARPA-E project on extracting terpene from pine trees for fuel. And there, you just tap the pine tree and you can just extract the sap, right? And so you can imagine, and that obviously requires further processing, but if we could really engineer plants with great control, you can imagine, not having to go to a traditional refinery like the ones that you have today at all. Maybe you can have more continuous processes where you're harvesting the good, for example, instead of cutting everything down and pulping it.
Thanks, Ben. Steve, this is a follow-up question, I think, to something earlier. Are you concerned that farmers would switch to biomanufacturing crops for increased profits, causing strain on our food supply chain? And maybe a similar concern would be with bioethanol.
Well, I guess, my response is that 40% of corn today is used for ethanol. Yeah. So we already are doing biomanufacturing on a large scale with corn. So I think this is more, can we be more thoughtful about it? I think the idea that we're trading off food and fuel or food and chemicals is a bit of a misnomer. I think we underestimate the ability of our farmers to get higher yields and our scientists to get higher yields from plants and accommodate both.
Thanks. Here's a question that comes in that's near and dear to my heart. It's about the water burden on each of these processes. And I wonder if each of you could address that in terms of what you presented today. We'll start with Zach.
Is there a water burden on anything I proposed?
For sure.
Which one? Well, the ocean. No, I actually, to be honest, I haven't really thought about that one. I guess maybe the microbe one, but wait, which ones have a large water demand?
Well, I think if you're looking at oxidation of these chemicals and you're thinking about photocatalysis, there's probably quite a bit of cleaning that would have to occur. Things build up on them. There's rinsing, washing, wastewater. I love wastewater, so I think about that a lot. Yep.
Sure, but actually these radicals are so reactive, right? That's actually what, what is it? Plasma cleaners do.
Yeah,
right? They actually pull things off. So
yeah.
I think the water use could actually be quite mitigated.
Okay,
yeah.
Cool. Yeah, thanks. Ben?
I'd say that there are many plant engineering traits which could alleviate water usage. For example, you can optimize deeper roots, maybe even make them perennial so that they can seek out water more efficiently, so they'll require less water. In addition, if you could introduce nitrogen fixation or enhance nitrogen use efficiency, you need less fertilizer, which leads to less of the waste that Charlie spoke about. But of course, if you rapidly scale up plant production without addressing the roots or the nitrogen, then you might need more fertilizer, which endangers the water system. So these things are all interconnected, but I think that there's great potential to use these tools to make a positive difference for water.
And also, perhaps with, genetic engineering, you can engineer for drought resistance, right?
Yes.
And so,
yeah.
So Steve?
This gives me an opportunity to talk about one of my favorite crops, sorghum. So sorghum is often grown is more drought tolerant than corn and is grown in areas west of the 100th meridian that have less water. So you can imagine, sorghum is compared to corn, there's only about 10 or 11 million metric tons of sorghum biomass. But you can imagine if we expand sorghum biomass or sorghum production and that we produce chemicals in sorghum, we may be able to overcome some of the needs, the problems that people are discussing with food, though I think there are less problems than people think, but also with resources like water, because we can use more drought-tolerant crops.
Yeah. All right. I think we're out of time. I really appreciate everyone's attention, and thank you all to the speakers.